Muscle twitch is when the muscle is stimulated with a single electric shock of sufficient voltage and it quickly contracts and relaxes. This consists of a latent contraction and relaxation phase. The latent period is when the shock produces an action potential that is conducted along the sarcolemma and stimulates the release of calcium from the sarcoplasmic reticulum. The contraction period is when calcium binds to troponin and stimulates the muscle twitch. The relaxation occurs. It will occur if a second stimulus doesn't result in summation. It's just the muscle relaxing again. Stimulus strength and motor unit response. Muscles respond to stimuli in a graded fashion. More force is generated for more demanding tasks, and forces increase in two ways. Summation, which is increasing the force of contraction of muscle fibers. Another stimulus piggybacks on the first twitch before it has fully relaxed. Summation is also increasing the frequency of stimulation of a muscle gradually. Increasing the frequency of stimulation of a muscle gradually increases the force of contraction, and incomplete sequestration of calcium is important. The second way forces increases in recruitment. This is increasing the number of muscle fibers contracting, the activation of additional and larger motor units to produce stronger contractions. A larger motor unit means it has more fibers attached to the motor neuron. Treppe. If a series of electrical shocks at maximal voltage is given to a fresh muscle so that each shock produces a separate twitch, each of the twitches evoked will be successively stronger up to a higher maximum. This demonstrates treppe, or the staircase effect. Treppe may represent a warm-up effect and is believed to be due to an increase in intracellular calcium, which is needed for muscle contraction. When athletes warm up, treppe, along with increased blood flow and temperature, help improve efficiency. How do we normally produce the smooth, sustained contractions of complete tetanus in vivo? By asynchronous activation of motor units. As different motor units fire at slightly different times, the contractions of some motor unit muscle fibers begin when other motor units fibers start relaxing, producing a smooth contraction of the whole muscle. Stimulus frequency and whole muscle contraction. The action potential lasts 10 milliseconds, or I don't know what exactly this means. Muscle contraction, 100 milliseconds. Oh yeah, the action potential will last for about 10. The muscle contraction could be 100 milliseconds. Muscles are always sensitive to stimuli, and the stimuli may come faster than a muscle's contraction cycle. High-frequency stimuli may cause a tetanus. Incomplete tetanus is when the stimulator is set to deliver an increasing frequency of electric shocks automatically. The relaxation time between successive twitches will get shorter and shorter as the strength of contraction increases in amplitude. This is sustained but jerky. Complete tetanus is when, at a particular fusion frequency of stimulation, there is no visible relaxation between successive twitches. Contraction is smooth and sustained as it normally is during normal muscle contraction in vivo. When the high frequency of stimulation is maintained, the muscle gradually loses its ability to maintain the contraction and fatigues. Increased calcium concentrations outside of the sarcoplasmic reticulum are important, so the cross-bridge cycle will continue. Types of muscle contractions. Number 1, isometric contractions. These involve an increase in tension, but no changes in muscle length during contraction. They're used to maintain posture. Muscle tension does not cause muscle shortening. Isotonic contractions occur when tension on the muscle is not equal to the force opposing the muscle. This results in changes in muscle length. This is used to move the skeleton. The two types of isotonic contractions are concentric contractions or shortening contractions. This is when muscle tension is greater than the force opposing the muscle and the muscle shortens, for example, curling an arm. Eccentric or lengthening contractions occur when the force opposing the muscle is greater than muscle tension, for example, uncurling the arm. What influences the strength of a muscle contraction? The number of active motor units, the number of active motor units, in parentheses, number of fibers in a muscle that will contract, the frequency of stimulation, the thickness of each muscle fiber, because thick fibers generate more force, and the initial length of muscle fibers. Length-tension relationship. The force required to prevent muscle from shortening is measured as the tension produced. This tension is maximal when the sarcomeres are at a length of 2.0 to 2.25 UM. This is the length of sarcomeres in a muscle at rest. When sarcomere lengths are greater than about 2.2 UM, the tension produced by the muscle contraction decreases with increasing sarcomere length due to fewer interactions of myosin and actin. Once they reach about 3.6 UM, there is no overlap of actin and myosin, and the muscle produces zero tension. This is like, when this happens, it's like the muscle was torn. When sarcomere length is less than 2.0 UM, the force generated by muscle contraction declines with decreasing sarcomere length due to cross-bridge action becoming less effective as the muscle fiber gets thicker and shorter. At a sarcomere length of about 1.25 UM, the muscle produces zero force or tension. Metabolism of skeletal muscles. Intensity of exercise can be measured by VO2 max, which is the maximum rate of oxygen consumption by cellular respiration, and it can be increased by training. This is also referred to as the aerobic capacity. It can also be measured by lactate threshold, which is the point during exercise when lactate rises significantly in the blood, typically around 50 to 70% VO2 max, and this is also called anaerobic threshold. Energy sources for muscle contraction. ATP is the primary source of energy, but stored ATP in muscle fibers can only power vigorous exercise for a few seconds. New ATP can be produced from phosphocreatine, fermentation, which is not anaerobic respiration, and aerobic respiration. Aerobic respiration is the metabolism of glucose/fatty acids in the presence of oxygen. It is the dominant mode of metabolism during light exercise. Increases in GLUT transporters and T-tubules of skeletal muscles increases the glucose uptake, and liver releases more glucose during exercise to compensate. Extra oxygen is stored by myoglobin in skeletal muscle. Fermentation. This generates ATP by metabolizing glucose without the use of oxygen, and it occurs above the lactate threshold. ATP is generated quickly but inefficiently, and the use of glycogen can power moderate and heavy exercise. The amount of stored glycogen helps determine the length of exercise possible. Lactic acid is produced and metabolized in the liver for additional energy. Phosphocreatine. This is synthesized during resting conditions. It's useful for high-intensity exercise, but not for endurance. It is converted to ATP by creatine kinase. It can be synthesized by the liver/kidney or obtained by diet. Oxygen debt and recovery oxygen consumption. After exercise, you continue to breathe heavily, and this excess oxygen helps to repay the oxygen debt. Oxygen is stored by hemoglobin in blood and myoglobin in muscle. The usage of oxygen in metabolizing lactic acid, or oxygen is used for metabolizing lactic acid by the liver. Slow and fast twitch fibers. The different types of fibers contain different types of myosin, different levels of myoglobin, varying blood supply, and are useful for different activities. Slow twitch or type 1 fibers are good at contracting relatively slowly. They are well supplied with blood. They do not fatigue as easily as type 2. There are more mitochondria. They have a high level of myoglobin. The myosin heads break down ATP slowly, and they are excellent for posture maintenance and endurance. Fast twitch or type 2 fibers contract rapidly. They have a less blood supply. They fatigue more easily, have reduced mitochondria, low levels of myoglobin. The myosin heads rapidly break down ATP, and they are excellent for rapid, intense movements. Muscle fatigue. Fatigue is the decreased capacity to do work and reduced efficiency of performance. Muscular fatigue is caused by excess potassium in extracellular fluids, which is relieved gradually, the depletion of glycogen, calcium accumulation in cytoplasm, and excess phosphate in cytoplasm, as well as lowered pH from lactic production. Central fatigue involves a possible reduced ability of neurons in the brain to signal to neurons in the spinal cord to produce movements. This often occurs before muscle fatigue sets in. Reflexes. The basic functional unit is the reflex arc, which is the simplest portion of nervous system capable of receiving a stimulus and responding. It consists of 1. A sensory receptor, 2. A sensory neuron, 3. An interneuron, but not always, 4. A motor neuron, and 5. An effector organ. And a reflex is an automatic response to a stimulus produced by a reflex arc. This may or may not involve the brain. Reflexes are homeostatic and can maintain blood pressure, carbon dioxide levels, and can prevent damage to the body. Monosynaptic reflexes have a sensory neuron directly synapsing with a motor neuron, and polysynaptic reflexes involve interneurons. The stretch reflex is also known as the knee-jerk reflex, and it's used to maintain balance as well. It requires the muscle spindle as a sensory receptor. Sensory neurons innervate the spindle and generate action potentials when the muscle and spindle are stretched. These neurons directly communicate with motor neurons in the spinal cord, which innervate the muscle containing the spindle. And then I have in parentheses monosynaptic reflex. I'm really not sure what that means. Stretch stimulates sensory neurons, which stimulates motor neurons in the spinal cord, resulting in rapid muscle contraction. The Golgi tendon reflex. This prevents excessive stretching of tendons. Requires the Golgi tendon organ as a sensory receptor, which is found adjacent to collagen fibers and tendons. The intense stretch stimulates action potential generation by these organs, which sends signals to the spinal cord and synapse with interneurons. Interneurons secrete inhibitory neurotransmitters, which block the activity of motor neurons controlling the muscle in question, leading to relaxation. Reciprocal innervation. For example, activating the stretch reflex in quadriceps causes the quads to flex and hamstrings to relax. Withdrawal reflex, rapid response to pain. Number 1, the pain receptors detect painful stimulus. 2, the sensory neurons conduct action potentials to the spinal cord. 3, the sensory neurons synapse with excitatory interneurons that synapse with alpha motor neurons. 4, excitation of alpha motor neurons results in contraction of flexor muscles and withdrawal of limb from painful stimulus. Crossed extensor reflex ensures the other side of the body responds as well as prevents loss of coordination/balance. This involves double reciprocal innervation. Smooth muscle. Smaller than skeletal muscle fibers, actin filaments are very long to allow for excessive stretching. There are no Z discs, instead dense bodies play a similar role. Single unit smooth muscle. Cells joined The cells are joined by gap junctions. Stimulation of some cells will cause others to contract. Some cells may function as pacemakers, and the muscle can contract on its own. Multi-unit smooth muscle, the individual cells must be stimulated by nerve fibers. Contraction of smooth muscle in both single and multi-unit. Calcium is important but plays a different role because smooth muscle lacks troponin. It can respond to hormones, action potentials, and neurotransmitter. The steps are: number 1, neurotransmitter hormone binds to G protein coupled receptor. 2, the alpha subunit binds GTP, interacts with calcium, and then interacts with the calcium channel. 3, the calcium channel opens and calcium floods into the cell. 4, calcium binds and activates protein called calmodulin, which then binds to an enzyme called myosin kinase and activates it. Myosin kinase transfers a phosphate to myosin, which can then interact with actin. Myosin phosphatase will remove this phosphate from myosin, leading to relaxation. And relaxation can occur slowly and maintain tension for some time. The whole contraction and relaxation process is slower for smooth muscle than in skeletal muscle.